Introduction to Microbiology. Introduction. The importance of microorganisms cannot be overemphasized. In terms of sheer number and mass, microbes contain an estimated 50% of the biological carbon and 90% of the biological nitrogen on Earth. They greatly exceed every other group of organisms on the planet. Furthermore, they are found everywhere, from geothermal vents in the ocean depths to the coldest Arctic ice. They also are a source of nutrients at the base of all ecological food webs. Most important, certain microorganisms carry out photosynthesis, rivaling plants in their role of capturing carbon dioxide and releasing oxygen into the atmosphere. Those microbes that inhabit humans are also important, helping the body digestion and producing vitamins B and K. Microbes are necessary for the production of bread, cheese, beer, antibiotics, vaccines, vitamins, enzymes, and many other products. Their ability to produce biofuels such as ethanol is also being intensively explored. History. Microbial disease undoubtedly play a major role in historical events such as the decline of the Roman Empire and the conquest of the New World. In 1347, plague, Black Death, and arthropod-borne disease struck Europe with brutal force, killing 1/3 of the population, about 25 million people, within 4 years. Over the next 80 years, the disease struck repeatedly, eventually wiping out 75% of the European population. The plague effects was so great that some historians believe it changed European culture and prepared their way for the reionization. Today, the struggle by microbiologists and others against killers such as AIDS. Scope of microbiology. It has a great impact on fields such as medicine, agriculture and food sciences, ecology, genetics, biochemistry, and molecular biology. The applied aspects are concerned with practical problems such as disease, water and wastewater treatment, food spoilage and food production, and industrial uses of microbes. Analyzing the relationship between microorganisms and human diseases, including the mechanisms of infection, immune responses, and the development of treatments and vaccines. Exploring the roles of microorganisms in ecosystems, biogeochemical cycles, environmental impacts, and their potential use in bioremediation. Investigating cutting-edge areas including antibiotic resistance, the human microbiome, genome editing, and their potential implications on various fields. Microbial taxonomy and classification. Classification of microorganisms. Important factor in classifying microorganisms is that some are composed of prokaryotic cells and other of eukaryotic cells. Prokaryotic cells, Greek pro, before, and karyon, nut or kernel. Organisms with a primordial nucleus have a much simpler morphology than eukaryotic cells and lack a true membrane delimited nucleus. In contrast, eukaryotic cells, Greek eu, true, and carrion, nut or kernel, have a membrane-enclosed nucleus. They are more complex morphologically and usually larger than prokaryotes. These observations eventually led to the development of the classification scheme that divided organisms into 5 kingdoms: the Monera, Protista, fungi, Animalia, and Plantae. Microorganisms, except for viruses and other acellular infectious agents, which have their own classification system, were placed in the first 3 kingdoms. Microbial classification. First, much has been learned about the detailed structure of microbial cells from the use of electron microscopy. Second, microbiologists have determined the biochemical and physiological characteristics of many different microorganisms. Third, the sequences of nucleic acids and proteins from a wide variety of organisms have been compared. The comparison of ribosomal RNA rRNA begun by Carl Woese in the 1970s was instrumental in demonstrating that there are two very different groups of prokaryotic organisms, bacteria and archaea, which had been classified together as Monera in the 5 kingdom system. Later study based on rRNA comparisons show that Protista is not a cohesive taxonomic unit and that it should be divided into 3 or more kingdoms. The 3 domains of life. Most microbiologists believe that organisms should be divided among 3 domains: bacteria, archaebacteria, and eukarya. The universal phylogenetic tree, as proposed by Norman Bass. The 3 domains of life represent the highest level of classification for all living organisms on the earth. They were proposed based on advancements in molecular biology and genetics, particularly the analysis of ribosomal RNA sequences. This picture showed that universal phylogenetic tree representing the 3 domains of life: bacteria, archaea, and eukarya. These evolutionary relationships are based on RNA sequence comparisons. The human genus Homo sapiens is highlighted in red. Cell structure and function. Prokaryotic microbial cell structure. Prokaryotes may be spherical, cocci, rod-shaped bacilli, spiral or filamentous. They may form buds and stalks, or they may have no characteristic shape, pleomorphic. Prokaryotes are much simpler structurally than eukaryotes, but they do have unique structures. Functions of prokaryotic organelles. Here we show all functions of prokaryotic organelles. Plasma membrane, selectively permeable barrier, mechanical boundary of cell, nutrient and waste transport, location of many metabolic processes like respiration and photosynthesis, detection of environmental cues for chemotaxis, gas vacuoles. It's necessary for buoyancy for floating in aquatic environments. Ribosomes for protein synthesis. Inclusion body for storage of carbon, phosphate and other substances. Nucleoid for localization of genetic material like DNA. Periplasmic space for contains hydrolytic enzymes and binding proteins for nutrient processing and uptake. Cell wall for provides shape and protection from osmotic stress. Capsule and slime layer. Its function for resistance to phagocytosis, adherence to surfaces. Fibrae and pili. Its function is attachment to surface, bacterial mating. Flagella. Its function is swimming, motility. Endospore. This is responsible for survival under harsh environmental conditions. Cell structure and function. Bacterial cell structure. The vast majority of prokaryotes have a cell wall outside the plasma membrane to give them shape and protect them from osmotic stress. Bacteria walls are chemically complex and usually contain peptidoglycan. Bacteria often are classified as either gram-positive or gram-negative based on differences in cell wall structure and their response to gram staining. Gram-positive walls have thick homogeneous layers of peptidoglycan and teichoic acids. Gram-negative bacteria have a thin peptidoglycan layer surrounded by a complex outer membrane containing lipopolysaccharides and other components. The mechanism of the gram strain is thought to depend on the peptidoglycan, which binds crystal violet tightly, preventing the loss of crystal violet during the ethanol wash. Archaea cell structure. Archaea cell wall do not contain peptidoglycan. Archaea exhibit great diversity in their cell wall makeup. Some archaea cell walls are composed of heteropolysaccharides, some are composed of glycoprotein, and some are composed of protein. Their wall chemistry varies from species to species, but usually consists of complex heteropolysaccharides. For example, methanobacteria and some other methane-generating archaea, methanogens, have walls containing pseudomurine. Cell structure and function. Eukaryotic microbial cell structure. The most obvious difference between eukaryotic and prokaryotic cells is in their use of membranes. Eukaryotic cells have membrane delimited nuclei, and membranes play a prominent part in the structure of many other organelles. Organelles are intercellular structures that perform specific functions in cells analogous to the functions of organs in the body. Functions of eukaryotic organelles. Plasma membrane. Its function is mechanical cell boundary, selective permeable barrier with transport system and mediated cell-cell interactions and adhesion to surface secretion. Cytoplasm. For environment for other organelles, location of many metabolic processes. Microfilaments, intermediate filaments and microtubules. Its function is cell structure and movements for the cytoskeleton. Endoplasmic reticulum. Its function is transport of materials, protein and lipid synthesis. Ribosomes. Its main function is protein synthesis. Golgi apparatus. It's used for packaging and secretion of materials for various purposes. Lysosome formation. Lysosomes. For intercellular digestion. Mitochondria. It is energy production through use of the tricarboxylic acid cycle. Electron transport, oxidative phosphorylation and other pathways. Chloroplast. It is used for photosynthesis, trapping light energy and formation of carbohydrate from CO2 and water. Nucleus. Its main function for repository for genetic information, control center for cell. Nucleolus for ribosomal RNA synthesis, ribosome construction. Cell wall and pellicle. It used for strengthen and give shape to the cell. Flagella. It play important role in cell movement. Vacuoles. It is important for temporary storage and transport, digestion, food vacuoles, water balance, contractile vacuoles. Comparison of prokaryotic and eukaryotic cells. Property. Organization of genetic material. True membrane bound nucleus. It is present in eukarya, either it's absent in bacteria and archaea. DNA complex with histones. It is absent in bacteria, either present in archaea and eukarya. Chromosomes. It is usually one cellular chromosome in archaea and bacteria, either present in more than one chromosome or linear in eukarya. Plasmids. Very common in bacteria and archaea, either rare in eukarya. Introns in genes. It is rare in both bacteria and archaea, either present in eukarya. Nucleus. It is absent in bacteria and archaea, either present in eukarya. Mitochondria. It is absent in bacteria and archaea, either present in eukarya. Chloroplasts. It is present in eukarya, either absent in bacteria and archaea. Plasma membrane lipids. In bacteria and eukarya, ester-linked phospholipid and sterols is present. Either bacteria have hapenoid is also present. In archaea, glycerol diether and diglycerol tetraether, some have sterols. Flagella. In bacteria, submicroscopic in size, composed of one protein fiber. In archaea, submicroscopic in size, composed of one protein fiber. Either in eukarya, microscopic in size, membrane bound, usually 20 microtubules in 9+2 pattern. Endoplasmic reticulum. It is present in eukarya, either absent in both bacteria and archaea. Golgi apparatus. It is present in eukarya, either absent in both bacteria and archaea. Peptidoglycan in cell walls. It is absent in archaea and eukarya, either present in bacteria. Ribosome size. It is similar in size in bacteria and archaea, about 70 s and 80 s in eukarya. Lysosome absent in both bacteria and archaea, either present in eukarya. Cytoskeleton. It is rudimentary in both bacteria and archaea, either present in eukarya. Gas vesicles. It is present in bacteria and archaea, either absent in eukarya. Cell structure and function. Cell functions, metabolism and energy production. Metabolism is the total of all chemical reactions that occur in cells. It can be divided into two parts. The energy conserving fueling reactions called catabolism and anabolism. Energy is the capacity to do work. Living cells carry out three major kinds of work: chemical work of biosynthesis, transport work, and mechanical work. Metabolic channeling localizes metabolites and enzymes in different parts of the cell and influences pathway activity. A common channeling mechanism is compartmentalization. ATP is a high energy molecule that serves as energy currency. It transports energy in a useful form from one reaction of location in a cell to another. ATP is rapidly synthesized from ADP and PI using energy released from exergonic reactions. When hydrolyzed back to ATP and PI, it releases the energy, which is used to drive endergonic reactions. This cycling of ATP with ADP and PI is called the cell's energy cycle. Microbial growth and control. Microbial growth curve. When microorganisms are grown in a batch culture, the resulting growth curve usually has four phases: lag, exponential, log, stationary, and death. In the exponential phase, the population number of cells undergoing binary fission doubles at a constant interval called the generation or doubling time. The mean growth rate constant K is the reciprocal of the generation time. Exponential growth is balanced growth, that is, cell egg components are synthesized at constant rate relative to one another. This picture shows microbial growth curve in a closed system. X-axis shows time and Y-axis show log number of viable cells. The four phases of the growth curve are lag phase, exponential phase, which is log, and stationary phase, and then death phase. Microbial growth and control. Control of microbial growth is very important. With help of sterilization and disinfection, we can control microbial growth. Sterilization. Sterilization is the process of completely eliminating all form of microbial life, including bacteria, viruses, spores, and fungi, from an object or environment. Methods of sterilization: heat sterilization, autoclaving, which moist heat under pressure, dry heat, and incineration. Chemical sterilization, using chemical agents like ethylene oxide gas and hydrogen peroxide vapor. Filtration, removal of microorganisms by passing liquids or gases through fine pores filters. Radiation, use of ionizing gamma rays, X-rays, or non-ionizing UV light. Radiation for sterilization. Disinfection. Disinfection is the process of reducing the number of pathogenic microorganisms to a level where they no longer pose a threat to public health but may not eliminate all types of microorganisms or spores. Methods of disinfection. Chemical disinfection, using chemical agents like chlorine, alcohols, phenols, or hydrogen peroxides to kill or inactivate microbes. UV disinfection, exploring microbes to UV light to disturb their DNA and prevent replication. Boiling, heat disinfection by boiling water or using steam for certain objects. Filtration, removing microorganisms through filters with pore sizes that trap pathogens. Microbial responses to environmental factors. Solute and water activity. osmotolerant, able to grow over wide range of water activity. For example, Streptococcus aureus. Halophile, requires high level of sodium chloride. For example, Halobacterium. pH. Acidophile, grow optimum between pH 0 to 5.5. For example, Sulfolobus. Neutrophile, grow optimum between pH 5.5 to 8. For example, E. coli. Alkalophile, grow optimum between pH 8 to 11.5. For example, Bacillus alkalophilus. Temperature. Psychrophile, grow at 0 degrees Celsius and has an optimum growth temperature of 15 degrees Celsius or lower. For example, Bacillus psychrophilus. Psychrotrophs can grow at 0 to 7 degrees Celsius, has an optimum between 20 and 30 degrees Celsius and maximum around 35 degrees Celsius. For example, Listeria monocystogenes. Mesophils has grow optimum around 20 to 45 degrees Celsius. For example, E. coli. Thermophile can grow at 55 degrees Celsius or higher. Optimum offer between 55 and 65 degrees Celsius. For example, Thermus aquaticus. Hyperthermophile has an optimum between 80 and about 113 degrees Celsius. For example, Sulfolobus. Oxygen concentration. Obligate aerobe, completely depend on atmospheric oxygen for growth. For example, Micrococcus luteus. Facultative anaerobe, does not require oxygen for growth but grows better in its presence. For example, E. coli. Aerotolerance anaerobe, grows equally well in presence or absence of oxygen. For example, Streptococcus. Obligate anaerobe, does not tolerate oxygen and dies in its presence. For example, Clostridium. Microaerophile, requires oxygen levels between 2 to 10% for growth and is damaged by atmospheric oxygen levels 20%. For example, Triphonema. Pallidum. Pressure. Barophile, growth more rapidly at high hydrostatic pressure. For example, Photobacterium. Profundum. Microbial diversity. Bacteria, the prokaryotic microorganisms. Archaea, extramorphile and unique characteristics. Fungi, yeasts, molds and mycorrhizae. Protists, a diverse group of eukaryotes. Viruses, obligate intracellular parasites. Bacteria, the prokaryotic microorganisms. It is diverse in shapes. Bacteria come in various shapes such as cocci, spherical, bacilli, dot-shaped, spirilla, spiral-shaped, vibrio, comma-shaped, and spirochetes, corkscrew-shaped. Antibiotic resistance. Due to the misuse and overuse of antibiotics, some bacteria have developed resistance, leading to the emergence of superbugs that are challenging to treat. Nitrogen fixation. Certain bacteria like Rhizobium form symbiotic relationships with leguminous plants, converting atmospheric nitrogen into a usable form, thus enriching the soil. Biotechnology applications. Bacteria are extensively used in biotechnology to produce insulin, enzymes, and other valuable products through genetic engineering. Biofilm formation. Bacteria can adhere to surface and form biofilm, which are complex communities of cells that provide protection and enable them to survive Harsh conditions. Archaea, extremophile and unique characteristics. Extremophiles, archaea are known for their ability to thrive in extreme environments like hot spring, thermophile, salt flats, halophiles, and acidic environments, acidophiles. Unique lipids. Archaea cell membranes consist of unique lipids called isoprenoids, which differ from the fatty acids found in bacteria and eukaryotic cell membrane. Methanogenesis. Methanogenic archaea produce methane gas as a metabolic byproduct during anaerobic decomposition, contributing to global methane emissions. Extensive diversity. Archaea exhibit a wide range of metabolic capabilities, playing important role in biogeochemical cycles and ecological niches. Early life on Earth. Some researchers propose that archaea might have been among the earliest life form of Earth, influencing the planet's early atmosphere. Fungi, yeast, molds, and mycorrhizae. Hyphal structure. Fungi grow as network of thread-like structure called hyphae, allowing them to secrete enzymes and absorb nutrients from their surroundings. Mycorrhizae. Mycorrhizal fungi form symbiotic relationships with plants, enhancing nutrient uptake such as phosphorus for both the fungi and the plants. Decomposers. Fungi are vital decomposer in ecosystems, breaking down dead organic matter and recycling nutrients back into the soil. Lichens. Lichens are a unique mutualistic association between fungi and algae or cyanobacteria, often found in harsh environments where neither organism could survive alone. Pathogenic fungi. Some fungi are pathogens, causing disease in plants, animals, and humans, like rusts, smolts, and fungal infections, athlete foot, and ringworm. Protists, a diverse group of eukaryotes. Diverse group. Protists are a diverse assemblage of eukaryotic microorganisms encompassing various unicellular and multicellular organisms. Phytoplanktons. Photosynthetic protists, known as phytoplankton, are the foundation of marine food chain and contribute significantly to global oxygen production. Malaria. Plasmodium, a protist parasite transmitted through mosquitoes, cause malaria, a disease responsible for significant morbidity and mortality worldwide. Amoeboid movement. Some protists like amoebae exhibit amoeboid movement, probably themselves using pseudopods. Symbiotic relationships. Some protists form symbiotic relationship with other organisms like zooxanthellae, living with coral reefs providing essential nutrients to corals. Viruses. Obligate intracellular parasites. Obligate intracellular parasites. Viruses cannot reproduce on their own and require a host cell to replicate and produce new viral particles. Viral structure. Virus consist of genetic material, DNA or RNA, enclosed in a protein coat called a capsid, which can be surrounded by an envelope in some viruses. Lytic and lysogenic cycles. Viruses can follow either a lytic cycle, immediate cell disruption, or a lysogenic cycle. Viral DNA integrates into the host genome. Viral evolution. High mutation rates and short generation times lead to rapid viral evolution, influencing disease and emergence and vaccine development. Phages and bacteriophages. Phages are viruses that infect bacteria, bacteriophages, and they have potential application in treating bacterial infections and controlling bacterial populations. Future prospect in microbiology. Advancements in microbial research, genomics and proteomics. Rapid advancements in DNA sequencing and protein analysis technologies will enable a deeper understanding of microbial genetics and fundamental capabilities. Metagenomics. Exploration of complex microbial communities through metagenomics will reveal hidden diversity and interactions, expanding our knowledge of ecosystems. Synthetic biology. The ability to engineer microbes for specific function will revolutionize biotechnology, medicine, and environmental applications. Microbiome studies. Further research into the human microbiome will lead to personalized medicine and novel treatments for various diseases. CRISPR and gene editing. Precise gene editing tools will allow targeted modification of microbial genomes, opening new possibilities for medical and industrial applications. Nanotechnology. Integration of nanotechnology with microbiology will lead to innovative diagnostic and drug delivery systems. Artificial intelligence. AI applications in microbiology will enhance data analysis, prediction of microbial behaviors, and the discovery of a novel antimicrobial compound. Impact of microbiology on various fields. Human health. Improved understanding of the microbiome will lead to personalized therapies, probiotics, and prebiotics to treat diseases and enhance overall health. Agriculture. Microbial biofertilizers, plant growth promoting rhizobacteria, and disease-resistant crops will boost agricultural productivity and reduce reliance on chemical inputs. Environmental conservation. Microbial bioremediation and wastewater treatment technologies will aid in environmental cleanup and pollution control. Biotechnology. Microbial enzyme and metabolic pathways will be harnessed for sustainable production of biofuels, bioplastics, and other valuable compounds. Food industry. Microbiology will ensure food safety through pathogen detection methods and extend shelf life through natural preservation techniques. Climate change. Microbial contributions to carbon sequestration and greenhouse gas reduction will be explored to combat climate change. Astrobiology. Understanding extremophiles and microbial survival in extreme environments will inform the search for life on other planets and moons. Conclusion. Recapitulation of key concepts. Microbial taxonomy. Microorganisms are classified into different groups based on their characteristics and evolutionary relationships, cell structure and genetics. Microbes' genetic makeup influences their functions and interactions. Microbial growth and control. Understanding how microorganisms grow and way to control their proliferation is crucial for various applications, including medicines and food safety. Microbial diversity. Microbes encompasses a wide range of organisms, including bacteria, archaea, fungi, protists, and viruses, each with distinct features and roles. Microbiology and human health. Microorganisms play essential role in both beneficial and harmful interactions with the human body, influencing our health and well-being. Microbiology and the environment. Microbes are vital components of ecosystems, contributing to nutrient cycling and environmental processes. Applied microbiology. Microbes have numerous practical applications, from industrial processes to agricultural practices and environmental remediation. Ongoing relevance and importance of microbiology. Medical advancements. Understanding microbial pathogenesis and immune responses leads to the development of better diagnostic tools, vaccines, and treatments for infectious diseases. Biotechnology and industry. Microbes serve as essential tool in biotechnology, contributing to the production of pharmaceuticals, enzymes, and biofuels. Agriculture and food safety. Microbial research aids in improving crop yield, promoting sustainable agriculture, and ensuring safe food production and preservation. Environmental sustainability. Microbiology play a critical role in monitoring and maintaining environmental health and addressing issues such as pollution and waste management. Technological innovations. Advances in microbiology such as CRISPR-Cas9 gene editing hold the promise of revolutionizing multiple scientific fields.